Approximately 3.8 million years ago, the African landscape consisted of diverse environments, including woodlands and open grasslands. Australopithecus inhabited these varied African environments. The mosaic of environments presented Australopithecus with diverse dietary challenges. Woodlands provided fruits, leaves, and insects as potential food sources. Grasslands offered seeds, roots, and tubers as alternative dietary options. Adapting to these dietary challenges was crucial for Australopithecus' survival in the African landscape. Climate fluctuations led to seasonal variations in resource availability. Periods of drought significantly impacted plant-based food sources. Reduced plant availability affected the diet of Australopithecus. Environmental changes created challenges for early hominid survival. Adaptation to climate shifts was crucial for Australopithecus. The primary plant foods consisted of tough grasses, seeds, roots, and tubers. These plant foods necessitated significant chewing capabilities. Effective digestion was essential to process these plant-based foods. Roots and tubers formed a crucial part of the diet. Fossil evidence indicates that Australopithecus teeth possessed thick enamel. Thick enamel suggests a diet that included hard and abrasive foods. The dietary adaptation of Australopithecus allowed them to exploit a wider range of food resources. Dental enamel thickness is a key indicator of dietary habits in fossil hominids. Analysis of fossil teeth provides insights into the evolutionary adaptations of early hominids. Australopithecus possessed a robust jaw structure adapted for processing tough vegetation. The jaw's strength allowed for efficient mastication of fibrous plant matter. This anatomical adaptation provided a significant dietary advantage. The robust jaw facilitated the consumption of a wider range of plant-based foods. This adaptation is a key feature in understanding the dietary habits of Australopithecus. Microscopic analysis of teeth provides direct evidence of past dietary habits. Phytoliths, microscopic plant particles, are identifiable markers of plant consumption. The presence of phytoliths on teeth confirms the consumption of grasses and other plant materials. Different types of phytoliths can be used to identify the specific plants consumed. Analyzing phytoliths provides valuable insights into ancient diets and agricultural practices. Dietary reconstruction through phytolith analysis enhances our understanding of past human behaviors. Isotopic analysis of bone collagen provides insights into dietary habits. Bone collagen isotopic signatures reflect the consumption of C3 and C4 plants. A mixed feeding strategy is indicated by the presence of both C3 and C4 plant isotopes in bone collagen. The relative abundance of carbon isotopes reveals dietary composition. During dry seasons, competition for food resources intensified among Australopithecus. Resource scarcity forced Australopithecus to adapt their feeding habits. Increased competition led to dietary diversification in Australopithecus populations. Adaptation to new food sources was crucial for survival during periods of environmental stress. Environmental pressures drove evolutionary changes in Australopithecus feeding strategies. Insects serve as an alternative protein source, supplementing a primarily plant-based diet. Termites and larvae are examples of insects consumed to supplement nutritional intake. Opportunistic feeding involves consuming available insects to enhance dietary protein. Insect consumption provides a valuable supplement to plant-based diets. Insects, such as termites and larvae, provide an alternative protein source. Early humans utilized simple stone tools to access underground tubers and roots. The use of stone tools allowed early humans to expand their dietary options. Accessing underground resources provided a more reliable food source. Simple stone tools were crucial for digging and extracting tubers and roots. Expanding dietary options contributed to the survival and development of early human populations. The ability to access underground resources provided a nutritional advantage. Group foraging enhances the ability to locate scattered food resources. Group foraging enhances the ability to exploit scattered food resources. Group foraging increases the efficiency of resource utilization across the landscape. Animals can locate and exploit scattered food resources across the landscape through group foraging. Group foraging provides a competitive advantage in resource-scarce environments. Enhanced foraging success through group cooperation ensures greater survival rates. Australopithecus exhibited dietary flexibility, enabling survival in diverse environments. Dietary adaptability allowed Australopithecus to exploit a wide range of food sources. This dietary flexibility was crucial for survival in variable and changing environments. Australopithecus's ability to consume various foods provided a significant evolutionary advantage. Exploiting multiple food sources ensured a more stable food supply for Australopithecus populations. Australopithecus likely possessed gut morphology distinct from modern humans. The gut structure of Australopithecus was adapted for more efficient digestion of tough plant fibers. Comparative analysis reveals differences in gut length and complexity between Australopithecus and Homo sapiens. Enhanced fiber digestion in Australopithecus supported a diet rich in fibrous plant material. Variations in gut morphology reflect adaptive strategies for different dietary niches. The ability to thrive in diverse habitats reflects an organism's capacity to adapt its diet to local food availability. Ecological niche is influenced by the range of habitats an organism can occupy. Dietary adaptation is a key factor enabling species to occupy a wider range of ecological niches. Local food availability directly impacts the dietary habits of species within a given habitat. Species adapt to different food sources to maximize survival and reproductive success in varying environments. Natural selection favors individuals with traits that enhance survival in specific environments. Traits that improve an organism's ability to obtain food are advantageous under harsh conditions. Efficient food processing enhances an organism's survival rate in resource-scarce environments. Individuals with beneficial traits are more likely to survive, reproduce, and pass on their genes. This process leads to the adaptation of populations to their environment over time. Natural selection is a key driver of evolutionary change, shaping the diversity of life on Earth. Australopithecus fossils' geographic distribution indicates successful dietary adaptations across different African regions. Fossil evidence suggests Australopithecus diets varied based on regional resource availability. The wide distribution of Australopithecus fossils implies adaptability to diverse environmental conditions. Dietary flexibility contributed to the widespread geographic range of Australopithecus. Geographic distribution patterns reveal how Australopithecus populations adapted to local food sources. Australopithecus fossil locations correlate with areas providing sufficient nutritional resources. Dental microwear pattern analysis provides insights into dietary habits. Microwear patterns reveal the types of foods regularly consumed. The abrasiveness of food directly influences the formation of specific microwear patterns. Analysis of microwear can indicate the hardness and texture of past diets. Reconstructing the paleo environment provides insights into the flora and fauna that coexisted with Australopithecus. Understanding the paleo environment helps to determine the ecological context in which Australopithecus lived and evolved. Fossil evidence, geological data, and comparative biology are used to reconstruct the paleo environment. The reconstruction of the paleo environment provides a more complete understanding of Australopithecus's place in evolutionary history. Periods of food scarcity likely resulted in nutritional stress. Nutritional stress can significantly impact growth. Nutritional stress can negatively affect reproductive success. Dietary stress presents significant nutritional challenges. Australopithecus may have practiced limited food storage as a behavioral adaptation. Food storage could have served as a buffer against seasonal food shortages. This behavior suggests a degree of foresight and planning in early hominids. Limited food storage represents an early example of adaptive strategies for survival. Australopithecus were vulnerable to predators while foraging. Predator vulnerability influenced Australopithecus feeding strategies. Habitat choices of Australopithecus were affected by the need to avoid predators. Foraging in groups provided a degree of protection against predators. Vigilance and awareness of surroundings were crucial for survival. The trade-off between food acquisition and predator avoidance shaped Australopithecus behavior. Chewing efficiency is critical for extracting nutrients from tough plant materials. The structure of the chewing apparatus directly impacts its ability to process plant matter effectively. Efficient chewing increases the surface area of food, facilitating digestion and nutrient absorption. Different chewing mechanisms exhibit varying levels of efficiency in processing diverse plant types. The force and motion generated during chewing determine the degree of plant cell rupture and nutrient liberation. Chewing efficiency is a key factor in determining the nutritional value derived from plant-based diets. The digestive system may utilize fermentation to process cellulose. Fermentation aids in breaking down plant fibers within the digestive system. Energy extraction from plant fibers is facilitated through fermentation. Fermentation enables the digestive system to extract nutrients from otherwise indigestible plant matter. Resource partitioning involves species adapting to utilize different resources to minimize direct competition. Dietary niche differentiation is a key aspect of resource partitioning among hominin species. Competition for resources can influence the feeding behavior and evolutionary adaptations of hominins. Resource partitioning can lead to specialized feeding strategies, reducing direct conflict between species. By occupying different dietary niches, hominin species could coexist in the same environment. Long-term climate changes gradually altered the landscape. Changes in climate impacted food availability. The evolutionary trajectory of Australopithecus was shaped by these environmental pressures. Adaptation to new food sources became crucial for survival. Daily energy needs are met through consistent access to high-calorie food sources. Foraging behavior is driven by the necessity to fulfill these daily energy requirements. High-calorie foods provide the necessary fuel for daily activities and survival. Consistent access to energy sources is crucial for maintaining metabolic functions. Energy balance is achieved when energy intake matches energy expenditure. Australopithecus, primarily herbivores, may have supplemented their diet by occasionally hunting small game. Hunting small game provided Australopithecus with essential nutrients not readily available in a purely herbivorous diet. The hunting of small animals likely involved simple tools and cooperative strategies among Australopithecus groups. Small game hunting represents an adaptive behavior that allowed Australopithecus to thrive in varied environmental conditions. This occasional hunting behavior provided a crucial evolutionary advantage for Australopithecus. Scavenging carcasses provided access to valuable protein. Scavenging carcasses provided access to valuable fat. Carcass utilization was especially important during lean times. Scavenging offered a crucial survival strategy. Carcasses provided essential nutrients when other food sources were scarce. Access to carcasses could significantly improve health and energy levels. Dietary diversity varies across different geographical regions. Regional dietary diversity reflects local food availability. Environmental conditions significantly influence dietary options in each region. Local food systems are closely tied to regional dietary habits. Dietary diversity is an important indicator of nutritional health and food security. Habitat selection is significantly determined by the availability of consistent food sources. Reliable water sources are a crucial factor influencing habitat selection. The presence of both food and water synergistically affects habitat suitability. Animals tend to select habitats that maximize access to essential resources. Consistent access to food and water ensures survival and reproductive success within a selected habitat. Social learning likely played a role in transmitting knowledge about edible plants. Efficient foraging techniques were also likely transmitted through social learning. Social learning facilitates the acquisition of essential survival skills related to food. Knowledge transfer through social interaction enhances survival rates in natural environments. Cognitive abilities enable the development of strategies for overcoming challenges. Problem-solving skills are crucial for efficient food acquisition. Advanced cognitive functions facilitate complex food processing techniques. Strategic thinking allows for optimized resource utilization in food-related tasks. Cognitive adaptations enhance survival rates through improved food management. Tooth enamel thickness correlates with the types of foods consumed. Increased consumption of abrasive foods leads to greater enamel wear. Enamel thickness is also directly related to the amount of wear experienced by the tooth. Foods with high acidity levels can accelerate enamel erosion, reducing its thickness. Variations in diet directly influence the structural integrity and longevity of tooth enamel. Trace element analysis of bone provides insights into dietary composition. Different food types leave distinct trace element signatures in bone tissue. The proportions of trace elements in bone reflect the relative consumption of different food groups. Analysis involves identifying and quantifying specific trace elements within the bone matrix. Strontium, zinc, and barium are examples of trace elements commonly analyzed in bone. Trace element ratios can reveal information about an individual's diet over time. Paleobotanical evidence aids in identifying plant species consumed by Australopithecus. Plant remains provide direct evidence of the vegetation available in Australopithecus's habitat. Analysis of plant fossils reveals specific nutritional resources utilized by early hominids. Microscopic analysis of fossilized plant tissues uncovers cellular structures and plant taxonomy. The study of fossilized pollen grains provides insights into past environmental conditions and plant diversity. Plant remains offer crucial insights into the dietary adaptations and ecological interactions of Australopithecus. Animal remains found at a paleozoological site indicate potential sources of meat. The presence of animal bones suggests opportunities for scavenging by early hominids. Analysis of animal remains provides insights into the dietary habits of prehistoric populations. Animal remains in archaeological contexts can reveal past ecological interactions. Taphonomy is the study of how organisms decay and become fossilized. Understanding taphonomic processes is crucial for interpreting the fossil record accurately. Taphonomic analysis helps reconstruct past diets by examining fossilized remains and associated evidence. Fossil preservation is influenced by factors such as sediment type, burial rate, and environmental conditions. Taphonomic processes can alter or destroy fossil evidence, leading to incomplete or biased records. Stable carbon isotope ratios in teeth and bone provide insights into dietary habits. The ratios of stable carbon isotopes indicate the relative proportions of C3 and C4 plants consumed. C3 and C4 plants have distinct carbon isotope signatures due to different photosynthetic pathways. Analyzing carbon isotope ratios helps reconstruct past diets and understand ecological relationships. Teeth and bone act as archives, preserving isotopic information over time. Variations in carbon isotope ratios reveal shifts in agricultural practices and food sources. Nitrogen isotope analysis provides insights into an organism's trophic level. The ratio of nitrogen isotopes indicates the proportion of animal protein in an organism's diet. Higher trophic levels typically exhibit greater concentrations of heavier nitrogen isotopes. Nitrogen isotope analysis is a valuable tool for ecological studies and dietary reconstructions. Strontium isotopes can be used to determine the geographic origin of individuals. Analysis of strontium isotopes in teeth or bones provides information about where an individual lived during the formation of those tissues. Different geographic locations have distinct strontium isotope ratios in the soil and water. Plants and animals incorporate these strontium isotopes from their environment into their tissues. By comparing the strontium isotope ratios in human remains to those of different regions, researchers can infer the individual's place of origin. Strontium isotope analysis can also reveal movement patterns, indicating where individuals migrated during their lives. Oxygen isotopes serve as tracers to identify the origin and history of water sources. The isotopic composition of water reflects the environmental conditions during its formation and circulation. Variations in oxygen isotope ratios provide insights into past climate conditions and hydrological processes. Analyzing oxygen isotopes helps determine the source of drinking water and manage water resources effectively. Isotopic analysis is a valuable tool for understanding water's journey and its interaction with the environment. Microweir texture analysis provides insights into the abrasiveness of consumed foods. Surface scratches on teeth reveal details about the hardness of ingested materials. Microweir analysis helps determine the types of food sources in ancient diets. The analysis of microweir textures provides valuable data on dietary habits. Macroweir analysis examines the overall shape and wear patterns of teeth. Tooth shape and wear patterns reflect long-term dietary habits. Different food types cause distinct macroweir patterns on teeth. Macroweir analysis provides insights into past diets and lifestyles. Wear facets and cusp rounding are key indicators in macroweir analysis. Advanced imaging techniques enhance the accuracy of macroweir assessments. Finite element analysis models the bite force during chewing. The model simulates stress distribution in the jaw during mastication. Finite element analysis visualizes bite force and stress distribution. The analysis provides insights into jaw biomechanics. This method is used to study jaw biomechanics and dental implant performance. Comparative genomics identifies genetic adaptations. These adaptations are related to digestion processes. Genetic adaptations also influence nutrient metabolism. Comparative genomics helps reveal the genetic basis of dietary adaptations. Understanding these adaptations provides insights into evolutionary biology and personalized nutrition. The gut microbiome's composition affects the efficiency of digestion. The gut microbiome's composition influences nutrient absorption. Different microbial communities lead to varied digestive outcomes. The gut microbiome is a complex ecosystem of microorganisms. The balance of gut microbiota is crucial for optimal digestive health. Fermentation processes in the gut facilitate the digestion of cellulose. Cellulose digestion through fermentation allows for the extraction of energy from plant fibers. Gut microorganisms play a crucial role in the fermentation and breakdown of cellulose. The fermentation process converts cellulose into simpler compounds, providing nutrients to the host organism. The small intestine's morphology directly impacts nutrient absorption efficiency from digested food. Intestinal villi increase the surface area available for nutrient absorption. Microvilli on the surface of villi further amplify the absorptive surface area. A healthy intestinal lining ensures optimal nutrient uptake. Nutrient absorption involves transport mechanisms across the intestinal cell membrane. Variations in intestinal morphology can affect individual nutrient absorption capabilities. Metabolic pathways convert nutrients into energy. These pathways support daily activities. Metabolic pathways are essential for survival. Nutrients are broken down through a series of chemical reactions. Energy is stored in the form of ATP, adenosine triphosphate. Different metabolic pathways process carbohydrates, fats, and proteins. Hormonal regulation plays a crucial role in controlling appetite. Satiety, the feeling of fullness, is also regulated by hormonal signals. Hormones influence foraging behavior, affecting the search for and acquisition of food. Food intake is directly impacted by hormonal control mechanisms. The interplay of various hormones ensures a balanced and regulated appetite. Behavioral ecology examines how animals optimize foraging strategies. Optimal foraging aims to maximize energy intake while minimizing energy expenditure. Foraging strategies are influenced by factors such as food availability and predator risk. Animals use different methods to find and get food efficiently. Successful foraging contributes to an animal's survival and reproductive success. Cognitive maps aid in remembering locations of food sources. Spatial memory helps in navigating the landscape. Cognitive maps and spatial memory are interconnected. Reliable food sources are essential for survival. Navigation skills increase chances of finding food. Spatial awareness is key to creating cognitive maps. Social cooperation enhances the ability to hunt larger game. Cooperative strategies allow for more effective defense against predators. Shared resources and coordinated actions increase survival rates. Social cooperation involves division of labor and specialized roles. Effective communication is crucial for successful social cooperation. Social cooperation leads to improved problem-solving and innovation. Stone tool technology broadened early humans' dietary options. Stone tools enabled access to previously unavailable food sources. Accessing new food sources contributed to improved nutrition and survival rates. The development of stone tools involved understanding material properties and applying specific techniques. Different stone tool types were developed for specific purposes, such as hunting, butchering, and processing plants. Cooking with fire may enhance food digestibility. The application of fire to food preparation can reduce the risk of foodborne illnesses. Heat from cooking denatures proteins, making them easier to digest. Cooking can break down complex carbohydrates into simpler sugars. The Maillard reaction, facilitated by cooking, improves flavor and aroma. Cooking gelatinizes starches, increasing their digestibility and palatability. Dietary specialization reduces competition for food resources among hominin species. Niche partitioning allows different hominin species to coexist by exploiting different food niches. Dietary adaptations, such as specialized teeth, support niche partitioning among hominins. Reduced competition through dietary specialization increases the survival rate of hominin populations. Dietary specialization is a key factor in the evolutionary success and diversification of hominins. Evolutionary trade-offs can occur between different physiological systems. Brain size and digestive capacity are two systems subject to evolutionary trade-offs. Increased brain size may necessitate a reduction in digestive capacity, and vice versa. Dietary choices can be influenced by evolutionary trade-offs between brain size and digestive capacity. Evolutionary pressures can drive organisms to optimize their dietary intake based on physiological constraints. Climatic variability imposes environmental stress on species. Environmental stress forces species to adapt their diet. Foraging behavior changes as a result of climatic variability and dietary adaptation. Species survival depends on successful adaptation to environmental changes. Dietary and behavioral adaptations are key strategies for coping with climatic variability. Resource depletion due to overexploitation can lead to dietary shifts. Overexploitation of resources can result in increased competition for remaining supplies. Dietary shifts are a consequence of resource depletion and altered availability. Increased competition arises from the struggle to secure dwindling resources. Migration patterns are significantly influenced by the availability of food resources. Suitable habitats also play a crucial role in determining migration routes and destinations. Range expansion occurs when populations move into new areas with favorable conditions. The search for food drives animals to migrate to regions with seasonal abundance. Migration patterns are not random. They are strategic responses to environmental pressures and opportunities. The interplay between food resources and habitat suitability shapes long-term migration trends. Population dynamics are limited by the environment's carrying capacity. The availability of food resources constrains population size. Environmental carrying capacity represents the maximum population size an environment can sustain indefinitely. When a population exceeds carrying capacity, resource depletion and environmental degradation occur. Food scarcity directly impacts population dynamics by limiting reproduction and increasing mortality. Disease ecology studies the effects of parasite infections. Parasite infections impact the health of their hosts. Parasite infections affect the nutritional status of their hosts. Disease ecology focuses on the interactions between parasites, hosts, and the environment. Understanding these impacts is crucial for disease management and conservation efforts. Skeletal morphology reflects locomotor adaptations. Limb proportions are indicative of foraging behavior. Skeletal structure provides insights into an organism's ecological niche. Bone length ratios correlate with speed and agility. Skeletal adaptations can be traced through evolutionary lineages. Analyzing skeletal morphology aids in understanding extinct species' lifestyles. Muscle attachments facilitate force exertion during activities like chewing and locomotion. Biomechanical analysis quantifies the forces generated by muscles during movement. Chewing involves complex muscle interactions to generate the necessary force for food breakdown. Locomotion relies on coordinated muscle attachments to produce movement and maintain balance. Biomechanical analysis of locomotion assesses joint angles, muscle activation patterns, and ground reaction forces. Muscle attachments and biomechanical principles are crucial for understanding movement efficiency and injury prevention. Joint mechanics influence the range of motion. Range of motion affects the ability to access food resources. Joint mechanics affect the ability to process food resources. Limited joint mobility can restrict access to essential nutrients. Efficient joint mechanics optimize food resource acquisition and processing. Bone density is directly related to nutritional status. The availability of calcium significantly impacts bone density. Essential nutrients other than calcium also contribute to bone density. Adequate nutrition is crucial for maintaining optimal bone density. Growth rates serve as indicators of nutritional intake during development. Developmental patterns provide insights into an organism's health throughout its growth stages. Consistent monitoring of growth rates and patterns aids in identifying potential health issues early on. Variations in growth rates can reflect environmental factors affecting development. Analyzing growth rates and developmental patterns helps optimize nutritional strategies. Reproductive success indicates an organism's fitness. Fitness is defined by the ability to pass on genes to future generations. High reproductive success correlates with greater genetic representation in subsequent populations. Organisms with traits that enhance survival and reproduction are considered more fit. Natural selection favors individuals with higher fitness, leading to evolutionary adaptation. Reproductive success is a key metric for assessing evolutionary fitness and adaptation. Mortality rates reflect the challenges faced in surviving and reproducing in a harsh environment. High mortality rates often correlate with limited resources and increased predation. Survival strategies evolve to counteract high mortality, including increased reproduction rates and enhanced defenses. Species adapt to harsh environments through various survival strategies to maintain population stability. Paleopathology identifies disease markers in skeletal remains. Disease markers provide insights into the health of past populations. Skeletal remains reveal information about lifestyle. Analysis of skeletal remains contributes to understanding ancient diseases. Paleopathological studies offer a glimpse into the lives and challenges of past societies. Dental caries can be indicative of sugar consumption. The presence of dental caries suggests a diet rich in sugary foods. Early hominins rarely exhibited dental caries due to their limited access to sugary foods. Sugar consumption directly contributes to the formation and progression of dental caries. The link between sugar and dental caries is a key factor in oral health. Enamel hypoplasia indicates periods of nutritional stress during tooth formation. Nutritional deficiencies can disrupt the normal development of enamel. Visible enamel defects provide a historical record of past nutritional imbalances. Enamel hypoplasia manifests as pits, grooves, or discoloration on the tooth surface. Tooth enamel development is highly sensitive to nutritional intake during childhood. Arthritis indiquait joint de génération. Joint degeneration may reflect the stresses of locomotion. Joint degeneration may reflect the stresses of food processing. Arthritis is associated with both physical stress and metabolic processes. Trauma and injury patterns offer insights into an individual's lifestyle. Analysis of injury patterns can reveal potential causes of death. Specific injury types correlate with particular activities or environmental factors. Understanding injury patterns aids in forensic investigations and reconstruction of events. Detailed examination of trauma can provide crucial information about the circumstances surrounding an incident. Australopithecus dietary adaptations enabled survival in harsh climate. These adaptations allowed them to exploit a range of food sources. Australopithecus adapted their behavior to environmental challenges. Dietary flexibility was crucial for thriving in changing environments. Australopithecus's success demonstrates the importance of adaptability in evolutionary survival. Please subscribe to the channel and hit the notification bell so that you will be updated for more videos.